Okay. Well, welcome everyone. Thanks for coming. We're here for Derek Wise's thesis defense. Any of you that were hoping that this was Derek Wise, the famous Canadian rapper, are likely to be pleasantly surprised, because actually this concerns Derek Lindblad Wise, the world traveler and prominent graduate student and renowned neuroscientist. And it's my job in this first couple of minutes just to tell you a little bit about Derek. Derek's going to tell you about his own scientific work, and so I won't preview that for you. But I will just say that Derek has been remarkable during his graduate career in a number of ways. First of all, he is just an absolutely incredible lab citizen and lab mentor. Some of his mentees have come from graduate school elsewhere to come be here today. And it's really a testament to what a great hands-on teacher Derek is. And we've really appreciated, both in Steve's lab and in my lab, having him around. But for those of you that didn't get a chance to interact with Derek, you may be aware of other aspects of Derek's interests and characters. He is really a nerd's nerd. He's interested in swashbuckling, sword and sorcery, fantasy novels, and video games, and Magic the Gathering, and things like that. And of course, he's interested in real sword fights. And the first lesson that he learned is never bring a stick to a sword fight. But he learned this lesson so well, actually, that he has, in fact, been an award-winning swordsman in the historical European martial arts world, small as it is. So this is just really a remarkable aspect of Derek that he's been carrying on while doing the scientific work that he'll describe to you, and hopefully he will go on in his scientific career to get the kind of accolades that he's gotten for his sword fighting. Of course, we will have this public defense and then questions, and then there'll be a private defense with Derek's thesis committee, including, I'd like to thank Dean Guanamano from UCLA. We had to make sure, because of the swords involved, that it was somebody remote, didn't feel physically threatened. But anyway, we will be examining Derek, and then either as a consolation or a celebration, we will be having a party on the first floor of the Shapiro Science Center, to which you're all invited. So with that, take it away, Derek.
All right, thanks so much, Sacha. So welcome, everyone, and welcome the remotes as well. I just want to make sure, again, before we start, because this is going to be the last time that I'm going to be thinking about.
We can see a presenter view.
Oh, yeah, you don't have to do it. Thanks. Do it every time, I guess. Rude. Okay, sounds good. So is the audio quality acceptable online? I see that Emma's video is good for me, so I can see that. Okay, great. All right. So let's begin. So my thesis work is on regards how a normally stabilizing mechanism in neuroscience can actually become destabilizing, can become a problem, leading to hyperexcitability of cells. So in order to discuss this, we have to start with a little bit of theory. I know, it is unfortunate, but it must be done. And so let us imagine that we have a little population of cells here, right? And the yellow ones will represent the ones that are active, right? And so if the gain through the network, which is like how information propagates through it, how the one cell becoming active causes another cell to become active, is at an appropriate level, we'll get the normal activity of the brain. You'll be able to think, you'll be able to process stimuli from outside, you'll be able to make decisions and act within the world, right? And you can see that the trace from an EEG of this looks fairly controlled and believable, right? However, if the gain is too low, the activity will fail to spread, and it'll die out within the system. And if activity is not going on in your brain, you're either in a coma or you're dead. So that is incredibly bad. And on the other hand, if the gain is too high, then activity will spread from the system out of control. All the cells will become active, and that is a scenario that we call a seizure, right? Everything being active at the same time also doesn't allow for all the complicated things that need to happen in the brain. So this homeostatic plasticity that I mentioned in my title slide is one of the mechanisms that keeps this all in check. When activity levels get too high for a given cell, homeostatic plasticity brings it back down. And when activity levels get too low, it'll bring it back up. So we can say from homeostatic plasticity that if it's to function correctly, it has to be reversible, bidirectional, and have a stabilizing effect. Okay? So it's especially important because learning in the brain involves two destabilizing processes. Long-term potentiation means that when cells fire at the same time, the connections between them strengthen. And led to its logical extreme, that means that the connections will strengthen and strengthen, absent any feedback. They'll strengthen and strengthen until they get to a point where all the cells are firing, leading to a problem, right? And likewise, the opposing process to this long-term depression means that when cells don't fire at the same time, they become less connected over time. Led to its logical extreme, that means that the activity will die out, and we'll get to our other extreme problem, right? So homeostatic plasticity then is the thing that stops both of these things from getting out of control. It makes sure that at least the firing rate of the cells remains within a constant band so that they can continue to give out input. Output is what I meant to say. So if we want to talk about homeostatic plasticity, we are at some point going to have to get into the mechanisms of it. And so I'm going to talk about the three really well-validated, huge ones that have been talked about a lot in the mammalian field. So first of all, we're going to talk about, during this talk, excitatory input from glutamatergic synapses, which are the most prominent type in the mammalian brain. We're also going to talk about inhibitory input from GABAergic synapses. The combination of these two, onto a given model cell will lead to more likelihood to fire from the excitatory inputs and less likelihood for that cell to fire from the inhibitory inputs. But all of this input is summated at the soma, which is the little cool triangle. And the input to output ratio might also be another important variable here that would determine how much output the firing that the cell will give. And so the intrinsic excitability is what we call this input to output ratio of cells. Okay, so the disorder of excitability that we're going to talk about today is epilepsy. And everyone knows what epilepsy is because it's quite common. It involves debilitating seizures. It's defined as being hyperactive and hyper-synchronous in the cells. And I'm sure you are all also aware that while it is debilitating during lifespan, it can also be fatal. So it's a pretty big deal, right? So it can come about in two different ways, or perhaps many more than two different ways, but two categories of ways that I want to talk about today, which is traumatic brain injury or other traumas that happen to the brain during life, like CNS infection or stroke. And you can see here that sometimes it actually takes quite a bit of time for seizures to develop after one of those initial inputs. So you can see that the 50% mark is by 2 years from that green line, 50% of people. Okay, so the other way that it can happen is genetic. And one of the really interesting parts about epilepsy is that somewhere between 24 and 40% of cases are idiopathic, which means that we do not know why they are caused, right? It could be that there's a constellation of smaller genetic causes that result in these people getting epilepsy, or it could be a number of smaller environmental factors that are not as big as a big traumatic incident that would be written down, right? And in general, between these two methods, there's actually an enormous vulnerability to epilepsy in the first year. It's about there's a rate of about 3 times as much during the first year to get epilepsy as there is for any other time up to 15. And this only goes up to 15, but that goes on for the rest of the lifespan until very old age. So there's this very odd developmental vulnerability to epilepsy that I will be exploring in more detail here, right? And the model that just explores this developmental vulnerability that we'll be talking about today, that links this developmental vulnerability to homeostatic plasticity, is that if you block activity in cells in the brain during this an early window of development, then animals develop lifetime seizures. The way that this is done is that a cannula injection of tetrodotoxin is applied to the animals, and then tetrodotoxin is continuously applied for several weeks. Now, tetrodotoxin is a voltage-gated sodium blocker. It's quite famous because it's in pufferfish, and it's the thing that will kill you in pufferfish if you don't be careful. Because all action potential activity is the result of these voltage-gated sodium channels, blocking those channels entirely blocks the cellular activity while the drug is present, right? So in this case, they start the treatment at an early age, 10 to 12 days. This P is going to be used throughout the talk, and it means postnatal day, or you could call it day, but we call it P. And by 10 or 12 days of this tetrodotoxin exposure, while the TTX is still being applied to the brain, by the way, while they're still silencing, seizures start to develop in these animals. And then they last for the entire rest of the lifespan in this sort of initial work, right? So this was done in the year 2000 by another group in rats, and then we, in our lab in an unpublished study, replicated it in mice. We just didn't get the end that we needed to actually publish, but. So notably, this has to start at P10 to P12. If you start this treatment significantly later, it doesn't have the same effect, and it doesn't cause lifetime seizures. I believe that they have some pre-seizure activity, but it's not. It doesn't actually develop into the syndrome. So from this, we get to the point where we have had limited deprivation, right? A period only during the beginning of development that leads to a lifetime disability, right? That's kind of odd. That's not a given, right? Not everything that you can do to an animal at an early age will lead to it being scarred for life. And in general, the brain is supposed to be able to heal from many things, and the fact that it doesn't is a big problem, right? So what's so special about this second week of development, this P10 to P12 time period? Well, it turns out that a lot is going on then. During development, the cells start off very simple with only a small number of very simple connections. And then over the first years of life, this is from human, but the same thing is true for mouse, over a shorter time span. They get more and more complex, growing into the arborized structure that you imagine for adult neurons, right? And so in this particular period in mouse, on the right-hand side, you can see this little bit down here says synapse initiation, maturation, and pruning. This is the time during which all of the synapses are getting set up in the brain, and it's also the time when axons and dendrites are becoming more complicated and taking their adult form, which the animal will need to in order to interact with the environment the way it needs to, right? So during this period, the idea is that you need feedback in order to obtain the right balance of all of these systems, because this is actually when it's getting set up. The other idea that might be interesting to think about is that homeostatic plasticity gain, in order to deal with all of these brand new synapses, might actually have to be very high. You might actually have to have a very strong homeostatic response, bringing activity levels back and forth as you set up these original synapses, right? So let's talk a little bit about how homeostatic plasticity could be maladaptive. This will be the rest of the talk now that I've set everything up, right? So let's imagine that we have a neuron or population of neurons that has a given firing rate, right? And while tetrodotoxin is applied to these samples, they're not going to be able to fire, right? That's going to be our model. And during this time period also, homeostatic plasticity is going to engage its deprivation-based response. There's too little activity, it's going to try to bring it back up. And so it's going to increase the strength of excitatory synapses, decrease the strength of inhibitory synapses, and increase the strength of intrinsic excitability. These are well characterized in the system, and we know that this is what happens during this period of full deprivation of activity, right? So when we release it from tetrodotoxin and allow them to fire again, all of these elements will be increased. And so when we allow the slice to do its normal, when we allow our population of cells to perform its normal activity, it's going to be hyperactive, right? Because all of these things are upregulated. But homeostatic plasticity is bidirectional, and so in theory, it might come back down and go back to a stable firing rate that's appropriate for the system. However, I have showed you that in some cases, if you do this for too long, you get to a place where it's not capable of reversing, it's irreversible, and so we're going to be looking for this case where it doesn't actually return back down to its baseline firing. Okay, so in order to do this, we need a model. It turns out that injecting things into the brain of an animal is a little bit difficult to do, and it also takes a long time. And so we're going to pursue a model. The model is called organotypic slice culture. We're going to take a young animal at postnatal day 7, and we're going to slice it in 300 micrometer increments into little slabs of tissue, which we can then grow on a membrane over medium, which will keep it healthy. And then at a certain point, we'll look at its activity using GCAM6F. I'll tell you more about that in a second. But once we know whether it's hyperactive or not, then we're going to follow up with other sorts of measurements that are a lot more nuanced and get to the mechanisms of what's going on, electrophysiology and imaging, which again, I'll tell you a lot more about in a minute. Okay, so that's the basic idea of the work. I want to tell you a little bit more about organotypic slice culture for the people who don't work with this system. So when you cut the brain in this way, you end up with these slabs of tissue, right, that come from the top of the brain, that go to the bottom of the brain, contain all of the stuff that was in that vertical plane, coronal plane, we call it in neuroscience. And within that, we maintain all of the cell types that were originally there. So if there are excitatory cells, those are there. Inhibitory cells are also there. The glia that support all of this work is there. And all of the connections between those cells that are preserved within the plane of our slice are preserved. Everything that is long range, that goes from this, the area that we're looking at, all the way across the brain, that'll be cut because there was literally a blade that went through it. And the last thing that's preserved that's unlike other model systems that you might use for this is the extracellular matrix. So the extracellular matrix has been studied a lot more in the last 20 or 30 years. It's very important for a lot of cellular functions, and it's maintained in the system where it isn't in a lot of other ones. Okay, so while slices are in this culture system, they actually regain a lot of the activity that they had before. At the moment that it's cut, the cells don't know what to do, they're not getting the input that they were getting before, but over time, they grow new connections with cells around them, and they begin to have very complicated activity. And so my outside committee member, Dean, this is his work from 2007, and it shows that these spontaneous events, the activity of these cultures returns and becomes more complicated and frequent over time, over the first couple of weeks of growth following the dissection. And at 10 days in vitro, DIV, we've shown the same thing. If you patch individual neurons, you can see them spike in these complicated activity patterns, which I'll tell you more about in a minute too. All right. So the model that I'm going to start off with is a fairly simple one. We're going to take our culture of cells, and the day after they're cut, we're going to add a little dot of virus to the top of the slice. And this dot of virus is going to express our calcium indicator GCAM6F, which you'll continue to see throughout the rest of it. After this protein starts to be expressed, it's expressed throughout the course of the rest of the study, and it normally reaches full potency at like 5 days or 10 days or something like that. So then, to some of our slices, we're also going to add tetrodotoxin, right, to the media, and when it's in the media, it'll spread to the slice itself, and it will silence the activity. We checked quite carefully to make sure that it actually does this. And in order to have a nice comparison to a normal slice, we're also going to look at an age-matched control. So we're going to be comparing the case where we have our slices exposed to tetrodotoxin for 5 days, to a case where they're exposed for 0 days, they're just doing their normal activity. Okay? And what we're going to be looking at, and then at day 17, after 5 days of TTX and the appropriate age-matched control, we're going to bring them into a spinning disc confocal microscope, and that microscope will allow us to look at this GCAM6F indicator. Okay, so what does the GCAM6F indicator look like? Well, it starts off as a dull green, and then when the cells begin to fire, it becomes quite a bit brighter. Calcium rushes into the cell, and when it does, it binds this indicator. And this indicator undergoes a conformational shift that allows it to become fluorescent where it was not fluorescent before. Basically, twists and becomes fluorescent. And that will allow us to see the increase in brightness, which we'll then be able to quantify in all sorts of different ways. But it would be a bad presentation if I didn't show you videos of this. And so, okay, so this is the control case. This is what happens when we haven't added anything to the slice. This is the case that it goes to on its own. And you can see that the cells light up and fire. It's kind of a complicated pattern that they follow. They're really doing quite a lot of different things. And you can see their whole structure as they light up. I think it's quite beautiful. However, when we look at the case where we've allowed tetrodotoxin to hit the slice, we'd expect it to be hyperactive, and indeed, we see that all of the cells come on at exactly the same time, and the calcium comes into them at the same time. And it's so bright that you can't even see the cells very well anymore. You can see them, you can see their little soma, but you can't see them very well. Okay, so in order to quantify this, we're going to start by defining a period where they're active, right? So these cultures, as I was showing you earlier from Dean's work, become spontaneously active in these periods where all the cells become active for a time and then they stop being active for another time. The time when they're active is called an upstate. So we're going to look in this period of time to see what the cells are doing in a number of different ways now. The first thing that we might want to know is how often are they active in this way. So when they're not treated with tetrodotoxin, the normal case, there's something like 1 or 2 per minute. In this case, the example has 2. And when they've been treated with tetrodotoxin for 5 days, they become profoundly more hyperactive, going up to 16. That's really a lot. And of course, it's statistically significant if we want to look at the graph, because it's a huge difference. But we might also want to ask, and I defined this at the beginning. We said epilepsy had to be hyperactive and it also had to be hypersynchronous to be a good model of epilepsy. And so let's look at whether they're synchronous or not. And so what we're going to do is we're going to take the pairwise correlation between each 2 cells during the upstate, and then we're just going to average them. How correlated are the cells on average across all of the upstates? You can see that this cell on the left has a correlation of about 0.5 during the period of this upstate. And that's because all of the cells are becoming somewhat active during this upstate. It is an upstate after all, that's what we're looking at. But when tetrodotoxin has been added for a period of time, correlation goes, just to remind everyone, from -1 to 1. So 0.98 is almost perfectly correlated, right? So it goes from somewhat correlated to perfectly correlated. All the cells are doing exactly the same thing at exactly the same time. And this is also statistically significant. And then the third thing that we're going to look at is whether each time that an upstate happens, whether it unfolds in the same way. And one way that we can look at that is we can look at one cell, which is each one of these gray lines, in different upstates, right? And we can see whether this one cell has its activity unfold in the same way in each one of the upstates. And then we can average that among all cells, among all upstates. So in this case, we see the control has a correlation of about 0.2, so it's less similar than one cell to another from before. But when tetrodotoxin is added, it unfolds in almost exactly the same way every time. 0.83 is also quite a high number. Okay. Also statistically significant. But in order for this to be a good model for our age-dependent problem that we saw, age-dependent lifetime disability that we saw before, we need to look at a broader range of ages, right? So we're going to start the TTX treatment after another 5 days in culture, going from P20 to 17 to, so we'll start with 12 to 17, then we'll look at 17 to 22, then we'll look at 22 to 27. So you're just starting it later now. And presumably, some of these are past the point where this critical development is happening to the same extent, right? And so we see, using our two best measures, this synchrony and stereotypy, that they become less synchronized and less stereotyped with the same treatment if you do it a little later, right? So I would conclude that this is modeling pretty well what we were seeing in the animals. I also want to show you, because it is an entire chapter in my thesis, but it's only one slide here because it is an entirely different story, but there are genes that constrain homeostatic plasticity, and this other work was looking at what happens when they're knocked out. And so in the absence of a constraining factor, the homeostatic response is much greater, right? And so what we saw here is, when you just look at a normal animal, we did this for a lesser period of time in TTX for complicated reasons that aren't worth getting into, but you can see that it shows the same behavior, it becomes hyperactive and hypersynchronous. But if these genes are knocked out in this case, it becomes incredibly hyperactive and still hypersynchronous. So it becomes even more active in some cases. You can imagine cases where homeostatic plasticity fails even more here, and that's also statistically significant. One odd thing about this, though, is that the knockout of these genes does not actually cause the cells at baseline without this tetrodotoxin exposure to work any fundamentally different. They actually are the same amount of excitable if they're allowed to go on their own way, which is kind of cool. If you want to learn more about that, you can actually read my thesis. So now we've established at this point that homeostatic plasticity can cause acute hyperactivity. This is not especially surprising, but we needed to make sure that this was the case in our model. So now we have to look at whether we can actually have the bidirectional recovery or whether that fails. So we might ask, does this change persist? So the experiment that we're going to do at this point is I'm going to start tetrodotoxin and I'm going to run it for 5 days, or I'm going to run it for a longer period of time, 10 days. You'll remember that in that original TTX cannula situation, that it took about 10 days for the seizures to develop, right? So that's why I'm targeting that amount of time. And in order to do this experiment right, we have to either start at the same day and end at different days, because it's developmentally regulated, or we have to start at different days and end on the same day. I tried both at different times, but the experiments that I'm about to show you right now are the ones where it ends at the same day. All right. So we're going to look at those acute effects, what happens right after we remove tetrodotoxin. And we're also going to look at the case where it's allowed to recover for several days to see whether it can, right? Whether the activity can recover with some time too. All right, so what you'll see is the control activity here in these experiments looks rather similar to what I showed you before. And when you look at the difference between adding TTX for 5 days or 10 days, if you look at it acutely, which is the middle to your right when it's released, both of them look profoundly hyperactive and profoundly hypersynchronous. It would be difficult for me to describe how they are different between those two cases, but suffice to say they're both extremely hyperactive. All right, and then, but given a chance to recover, it looks like in this example case that 5 days of TTX recovers quite a lot more. It looks a lot more like the original case, just visually, right? But now let's look at it quantitatively. So let's look at that synchrony measure. So on the left, so I'm going to explain this in a little bit of detail because I'm going to use it a lot. On the left, you're going to see when we remove TTX acutely and the age-matching control for that time period, that the 10 days of TTX is profoundly synchronous, and the 5 days of TTX is a little less so, right? And by given a chance to recover, the 10 days of TTX recovers a little bit, not enough to be statistically significant from what it was before, and the 5 days of TTX recovers a little bit more, but not a whole lot, right? So in both of these cases, we can say it became hyperactive and then it didn't fully recover, right? Statistically also. And then a similar thing is true for the synchrony measure, the autocorrelation. But in this case, it looks like the 5 days of TTX actually does recover on this way of measuring hyperactivity all the way back down to baseline. So it's complicated as to exactly why all of these things are a little different from one another. But we can conclude that both durations of silencing do produce acute epileptiform activity, and 10 days of TTX appears to become more epileptiform and is less recoverable. Right? So we've got a progressive shift. We might also want to be interested, like we were showing before, in measuring the number of these upstates. And it looks like the 5 days of TTX actually displays, on average, more of these upstates than the 10 days of TTX does, right? It's the other order. Both of them become hyperactive, right, by this measure. And both of them recover somewhat, but not all the way back down to the baseline. And if we're going to look at the duration of these upstates, similarly, the duration of both of them becomes quite a bit shorter. You know, it's these brief bursts instead of this long, extended upstate. But and both of them recover somewhat on that measurement. All right. So we can conclude that both durations are hyperactive, and 10 days of TTX is slightly less frequent. But I'm going to pause for a moment here because this is a little bit discordant with what I just said a moment ago. It's discordant because hyperactivity measured in this way, if the frequency of these upstate periods, doesn't also take into account something else that's a little bit harder to measure, which is in each one of these upstate periods, how much activity is there inside of it? How would you measure that? You'd patch each one of the individual cells, record from them electrically, and figure out how much they burst during these periods, but I haven't actually done that experiment. So my conclusion is that the measures of synchrony are much more important. That's the question about whether it's a seizure or not a seizure, and the question of how often the seizures occur is less important to whether there's a seizure or not. Does that make sense? So we come down to the conclusion that 10 days of TTX causes a more profound disruption in these networks than does 5 days of TTX. And now we get to the promised question that I told you I would certainly be looking at, which is, okay, so like what's actually going on in these cells that's causing this problem, right, in this model network that we have here, right? So I'm going to look at what is different with the 10 days of TTX condition allowed a chance to recover, and the 5 days of TTX condition allowed a chance to recover, because those are the ones that demonstrate whether there's a profound, like analogy to lifelong disability, whether there's something wrong that will persist over time. Also, it's half the amount of work as doing it at both time points. So we're going to look at the same three different mechanistic components of the circuit that I was talking about before that have been well described for homeostatic plasticity. So we're going to be looking at excitatory synapses, inhibitory synapses, and the intrinsic excitability of the cell with the same logic. And we know that deprivation-induced homeostasis, as I was saying before, increases the strength of excitatory synapses, decreases the strength of inhibitory synapses, and increases the strength of intrinsic excitability. Now, we know that happens during the time that it's silenced. But it's a different question to say which of these things remains a problem after there is an opportunity to have the recovery, right? So that's really what we're looking at. We know that this happens immediately during the time, but we want to know which one stays permanent, okay? First, we're going to look at this intrinsic excitability, and the way that neuroscientists often do this is with the FI curve. So if you have your little cells in a dish, you drop an electrode down to them, and you make a seal so that you can record the electrical activity within a cell, right? When you do this, you can keep that cell around for 10, 20 minutes, an hour, a couple hours, depending on how good you are and how good your seal is, and you can record the electrical activity and you can give it stimulations. In this case, we're going to try to give it current stimulation, right, which you can see is the set of little current steps that I've marked off as little bars. And as we increase that current stimulation, see 50, 200, and 400 picograms, the cell starts not firing at all because we haven't given it enough input for it to fire an action potential, but then it begins to fire more and more as you add more and more current. And then we quantify this whole thing as a nice little curve where it's the relationship between the current that we've injected and the firing that it produced, right? So now let's look at our actual conditions. So I want to draw your attention mostly to the bottom left, where we look at that curve that I just described for you, which shows that the 10 days of TTX in purple is significantly elevated from the black baseline. The red 5 days of TTX is also elevated somewhat, but it fails to meet statistical significance for any values. The current times condition ANOVA test interaction is significant. And that's the most important thing to take away from this. So there is an impact of current, of the condition on these current steps statistically. And in order to do these ANOVA tests, what you then do is you do a series of stringent post hoc comparisons between each two, at each one of your comparisons. And in this case, 2 of them emerge from that stringent analysis as being statistically significant from baseline. Another property that really affects this intrinsic excitability practically, one of the cellular bases for the intrinsic excitability is called input resistance, and it's basically how much of the current that you put into the cell is retained, how leaky the cell is. And this increases dramatically over the course of our deprivation treatment. And then the last thing that we might be interested in is when do these cells start to fire? When you give them a sort of current injection, when do they start to fire? And that doesn't really have a large change. Okay. So we conclude then that there's an increase in this intrinsic excitability measure, and also which is paired with an increase in the input resistance of the cells, which I'll come back to in just a bit. Okay. So we looked at intrinsic excitability, and now we're going to look at the synapses. We're going to look at the synapses in 2 ways. One of them is that we're just going to take a picture of them, and we do this on a super resolution confocal microscope because our organotypic slice cultures are quite thick and dense with these synapses, and they're very small. And the logic of this is basically that we have 2 components that we're looking at when they come together. We have the presynapse, which is the axonal bouton, which is where one cell sends its signal all the way down, and then it's received here. And we're going to label that presynapse with one of 2 colors, either a far red or a red color, right? And then we're going to look at its connection on the other side at the postsynapse, and we're going to label that one green in this example here, right? And so when these things are together, we infer that there is a synapse there, because if there's a presynapse and a postsynapse together, that's a synapse. And what that actually looks like in practice is here, where I've blown up one of the little examples of a full synapse here, and then a bunch of other ones are circled. Okay. So, in order to do this synapse imaging, it actually took quite a lot of time, and I'd like to thank specifically Emma, Sam, and Catherine, who did a lot of really grueling work on this. You know, like we needed to figure out how we should threshold our little dots from the background noise. We needed to figure out how to deal with the fact that as you go deeper into the tissue, our signal degrades, and we also needed to figure out how we could sharpen our images further, because again, these things are very small and it's kind of fuzzy. But suffice to say, we came to a solution that we're fairly happy with. We may keep iterating on it, but we're fairly happy with it, and then I can show you how many of these synapses are and how big they are. So in this case, we're going to look at excitatory synapses first, and in this case, I've colored the presynapse, which is tagged with bassoon. Don't worry about exactly what that protein does, with a magenta, and the postsynapse with PSD95, which is excitatory specific, in green. So again, when these 2 things are in the same spot, then we'll infer that there's a synapse there. And you can see here, looking at the images that I've obtained in this way, in kind of a rough way, that it looks like there's somewhat of a decrease in the number of them. You can see the density of these connections becomes somewhat lower. And it looks like maybe they're a little bigger, but it's hard to tell for real. But that's why we quantify. So now let's quantify. So the synapse density is on the left here. And you see that there is a small decrease, but because there's a range of values, it's variable. It fails to meet statistical significance here. And it looks like there's a small increase in the size of these compartments, both halves. But because they're a little noisy, neither one of those fails. Both of those fail to meet statistical significance as well. And so we conclude that there is no large change in the excitatory synapses looked at in this way. All right. So the other way that we're going to look at synapses, aside from just taking a picture of them, is we're going to look at their the functional electrical activity that they have at the level of the cell. In order to do this, we're going to take a recording electrode like we did before, and we're going to patch it onto the cell body of the cell. And once we have this connection established, we're going to be observing little signals. So each one of the synaptic contacts that a cell has, when an action potential comes through, will release a large number of vesicles that will then be picked up by the receptors on the postsynaptic side of the cell. However, the way that this works is by a probabilistic method. Normally, when the action potential is not coming through, the probability of this event is very low. And when the action potential comes through, it goes up to an incredibly large number, like 1 million per second. It's that kind of probability range, right? So in this case, we're just going to be looking at those small, spontaneous events, those low probability things. But they'll allow us to understand how many receptors are on the postsynaptic side and how many of these synapses exist, because we know how much is being released, and so we could know how much what that corresponds to in terms of strength. So we're going to get these little traces that have these little sharp current inputs into the cell, and those are the minis, is what we call them. We can look at their amplitude, right, which tells us how strong the synapse is, and we can also count them over a period of time. I'm going to get into this in a little bit more. I'm going to explain this now so that I can use it in a moment, but the frequency of minis actually concords to 3 different things, or maybe even more, which is how many synapses contribute to that estimate of probability, what the probability is for each one of the vesicles that is at each one of the synapses, and also I said how many, and then what the probability of release is for each one of them, right? And the multiplicative combination of these 3 will result in the frequency, right? Okay. So we've got some example traces here of my recordings, and we can see that for the excitatory cells, these minis increase just a little bit, like 20% on the average mini that I've made from 100 example minis put together. And the amplitude of these minis, as you can see when quantified, results in a non-significant increase, but a small increase again. And the frequency of them doesn't have a large change either. Okay? So taken together, we conclude that there's not really a significant change in excitatory synapses based on either the imaging or the minis. Now, of course, there was a change as tetrodotoxin was being applied, but the argument is that it has recovered since. Right? Okay. So now let's look at inhibitory synapses in the same way. I'm going to go a little faster now because I've explained how it's been done. We have a presynaptic marker here called VGAT, which is related to GABA, and another one, Gefferin, which is related to the GABA receptor. And you can see here that in the example pictures, there's a pretty stark decrease in the density of these objects, right? You can see it with your eyeballs. It's quite real. And indeed, when we quantify it, it's quite real. I think it's something like down to 30%. And so there's a large decrease in the number of inhibitory synapses in our cultures when they're allowed, when they're put through this treatment. Okay. And it looks like on the right, maybe there's a small increase in the size of these compartments, but again, it fails to reach statistical significance when we quantify it. Okay, so then we're going to look at the minis. For the minis for inhibitory synapses, they get significantly bigger here, and the frequency does not change. But now I have to explain a little bit about how this could possibly be, because to the initiated, this is actually a contradiction. So I have said that there's a change in density, right, a decrease in density, but the frequency of the mini iPSCs does not change, right? So how can there be the same number of functional events if there are fewer synapses? Well, it's because there can be compensatory effects in the other 2 elements that contribute to this frequency, like I described before, and I bet that's what's happening. Okay. Hard to know for absolute certain because those measurements are actually a lot more difficult to make, but they can be made with electrophysiology. Likewise, it's a little bit discordant that we see a change in mini iPSC amplitude, but the actual volume that we observed in the picture is not increased significantly. And my view on this is that the change that we see in the volume is too small to see, but it does actually reflect a functional change that is statistically visible when we actually look at it with the mini measurement. Okay. So I think I'm a little low on time, so I'm going to skip what this means precisely. So silencing, then, we can say, causes a worsening hyperactivity. Right? When we add silencing for a longer period of time, we get to a state where it's profoundly hyperactive and it's not going to go anywhere. We see that there are going to be fewer inhibitory synapses and bigger inhibitory minis. So that's concerning. And greater intrinsic excitability and greater input resistance, right? And alongside this, of course, we said that there were smaller excitatory synapses that do not meet our threshold for making conclusions, right? So now let's talk about the potential circuit that's left behind for just a minute in broad terms without too much specificity. So if we imagine a population of cells, I'm going to mark that they have increased intrinsic excitability with this little red bar near the axon initial segment. And as we move from this small population of neurons to the next population of neurons, the next node in our little circuit, the connections from excitatory cells are going to be relatively close to what they were before, and the connections for inhibitory cells are going to be less in general than they were before. And so more of the next ones are going to be active in this second node than were active in the first node, and so on as we continue throughout the circuit, right? So this is the problem that we saw before. If the gain in the system is too high, activity spreads out of control, and you get a network deformed system. So then let's just finish up with the general conclusions. So homeostatic plasticity is supposed to stabilize activity, right? I said that it had to be reversible, bidirectional, and it had to be stabilizing in its overall effect. Right? But what we see here is when we add tetrodotoxin for a long period of time, we get this deprivation-induced response that mounts and mounts. And of course, this has to happen during a certain period of development. When it's released from this deprivation, then it has an opportunity to homeostatically readjust. But either something is fundamentally broken about the bidirectional nature of homeostatic plasticity, or it's just that the effect is the same size, but it's trying to repair a problem that is too big, right? And so we do not see it come back down to baseline. So, and it also might be that early on in development, in this way, that the gain of homeostatic plasticity is much larger than it would be at another time. And so maybe even the slope of homeostatic plasticity is extremely high at this time. That's speculative. So then we come to this conclusion. Homeostatic plasticity is required for stability. We know this. We've shown this in many ways. It's required in order to have this learning system. But it also has a dark side, right? And the dark side is that if you make this little program in the cell that adjusts activity levels up and down, you need to troubleshoot it for all of the possible scenarios that it could encounter in the world, right? And each one of those things is costly evolutionarily. You make a trade-off by making these extra bits of code. You only have a certain amount of code, right? And so during this period of profound synaptogenesis, profound change in the brain, when it's all getting set up, it seems like there are too many things going on for homeostatic plasticity to be able to adjust here. And maybe you just get to the place where it is mechanistically simpler and more efficient for the organism in general, for some animals to get epilepsy if they run into a big problem when they're young, so that you can have all of these other functions really work. And then the last thing I want to talk about real quick is how homeostatic plasticity might be misapplied in purpose, in practice, in two different examples, right? I said that I said that epilepsy can happen through traumatic brain injury or other events that are really traumatic, and I said that it could happen genetically. So I'll give you an example of both of those things. So the example for TBI is one that I've been thinking about for a long time, but it's not really proven. But imagine that you get an impact to the brain. We know that cells in this area die, and so the cells around them are going to be missing some of the inputs that they had before, making it so that they'll have less activity. But then also, we know that another thing happens when cells die. They spill out their internal components into the extracellular space, and all of the other cells are bathed in them. And the cells that survive come into contact with one component in particular that's really important, which is this glutamate, the glutamatergic synapse, the excitatory synapse, the neurotransmitter that comes out of this and causes a signal. Cells know that when there's too much of a concentration of this, it means that something's gone wrong. And so these connections silence, and therefore, while this extracellular glutamate is present, there's something called synaptic silencing that happens at each one of these synapses. So these cells might, for several reasons at once, become silent for a period of time, causing this deprivation-induced response that might be analogous to what we're talking about here. The other thing is that if you have genes that are responsible for the hyperactivity-induced response and the deprivation-induced response from homeostatic plasticity, if these things break so they're less effective, for example, in the hyperactivity-induced response, if you get a loss-of-function mutation that causes a weaker response, then you might get hyperexcitability like we're seeing here, right? So a person could develop it in that way. And also, for a deprivation-induced response, maybe you have too much of a response or the response doesn't shut off at the right time, and you can get hyperexcitability in the same way there. And that is all I have time for. So I will call it an end and ask questions.
Nice talk, Derek. So your hypothesis, if I understand correctly, is that when you assess neural activities like during an early, like in life, will cause like lifelong, like epilepsy. But in your last cultures, you treated the culture from P12 all the way until like 10 days until before you record. So have you tried just 5 days from P12 to P17 and withdraw the drug, and then see you can still see the same effect later?
Yeah. So
I couldn't hear the question.
If you start TTX at the same time at P12 and only go for 5 days, will you see the same effect that I was showing if you, well, the actual comparison is inside the work that I was doing if you start at 17 and go to 22 with the same duration of TTX silencing, right? So basically it's the question of like, what if you started at a different time and ended at a different time? Sorry, started at the same time and ended at a different time, as opposed to starting at a different time and ending at the same time, right? So we did try that and it had similar effects is the short answer. I just don't have room to talk about everything that I did. So, hey, Tom.
Hey, on a similar vein, we could see that at, I think P25, the 5 TTX space had somewhat recovered, but the 10 had not. But it looked like from your curves of synchrony and hyperactivity that it was still decreasing. Did you look at maybe like at what point it could possibly recover from the 10 days of TTX?
Yeah. This is the other obvious, this is the other big question that is obvious is a little rude. Sorry. The other big question that is left open by what I showed you today, I did look longer. I waited for it to go for 10 days. I could, you could expand that experiment as long as you wanted, right? You could go to 40 days or whatever. But I went to 10 days, and by that point, it had not recovered. In fact, it's this one. By that point, it had still not recovered in terms of its cross-correlation and autocorrelation. Weirdly, it does in terms of its frequency, but as I was saying, that is actually a less important measure in my take on this work. Hey.
So you did a very extreme perturbation. And the question I would have, do you expect that if you did milder perturbations, which is probably more like what most of us have going on in our brains, whether there'd be some sort of a graded amount of recovery, or whether there's some amount of perturbation that you can get complete recovery, and then there's a threshold over which now things really start going awry?
Yeah, I think I can answer it in two different ways. So one of them is like, so this is an extreme perturbation in two ways, right? One of them is that it's absolute silencing, right? And then the other one is that it's quite So the
It's
absolute...
not long, but it is absolute silencing.
Yeah, well, I mean, it seems like it's long enough, I guess, right, to cause a problem. But so for the absolute silencing question, I do imagine that there is an amount of activity that you could allow back into the circuit and still cause a problem. But I would bet that the cells can compensate for quite a lot of change, right?
It'd be really nice to
Yeah.
figure out a way to find that threshold.
Well, the problem with that is most of the changes that I can think of are pretty strong. Like if you add a drug to the culture, right, you can block a certain percentage of the receptors, but then the cells can upregulate the remainder, so they can just have more of them. And of course, maybe that is indeed what we're looking at. Anyway, I guess the short version of this is, I would imagine that if you leave even a little bit of activity, that the cells are probably able to compensate pretty well over a pretty large range. You know, there's a lot of work that says that these circuits are incredibly resilient to change and can find many different solutions, right? Your work. And so I think that's what would happen in general. Hey.
Hey Derek, great job. I had 2 quick questions for you. The first is, I think I missed where in the brain you were doing this.
Oh, that's a good point. I think I didn't mention actually.
So I was wondering how unique to this specific location your results are, versus if it's like a general thing or a local thing.
So we do this in cortex, and specifically the calcium imaging was in layer 2, 3, and the rest of it was in layer 5 for complicated reasons, which I can talk about more to the committee. But how general is it? So it probably isn't completely general, but the thing about activity level compensation is that it has to be a general rule for all of the cells in the brain in at least some sense, because the way that cells communicate is based on their activity and the propagation of their activity to another cell. And so it has to be general in at least some sense, right? So I don't know if we'd see exactly the same response, whether other areas of the brain would be more resilient to this, but in general it should be a law. There was another question.
Yeah, I was also wondering how different you think your results would be if you would have done like P0 or juvenile AAV injections instead of doing this in the slice culture.
Yeah, so the problem with those is that if you want to do that over this time period, you have to find a way to silence cells. And the way that makes the most sense is designer receptor is activated by designer drugs, DREADs. And that sounds like a great experiment to me too. I bet that this time period is quite special, but maybe if you go a little earlier, it's also really bad. If that answers it fully. But you can't really do exactly the same experiment. Hey, Ronnie.
Good talk. So I have a question with regards to the final hypothesis that, or rather interpretation you arrived at, is that you think that the slope of the homeostatic change is just not high enough for it to recover completely in the time that you're looking at it, or potentially never. So do you think something like triggering an opposite homeostatic response, so like inundating the circuit with activity, would be able to bring it back to baseline?
Yeah, I think that that's quite possible, but there is one problem that comes up from the results that I actually have here, which is that if you lose inhibitory synapses and they compensate to become larger, then you might actually not have the tools to do this in the way that you need to anyway, right? So because you have, that was the slide that I skipped, but if you have fewer inhibitory synapses and all of them are larger, you don't have the same fine-tuning ability. So it is quite possible that you could induce a homeostatic change in the other direction, like within the cell, activating the homeostatic transcription factors that we know about, but I imagine you would also need to do something about these other changes, right? Which I would argue is one of the reasons why it's so important to do work like this, because otherwise you wouldn't know all the things that you need to hit, right? Yes, Steve?
So Derek, you had one question, I think, from Mark Zielinski in Zoom. If you go to the podium computer, you can look at the chat.
Okay, you guys can't see this good. Okay, question for the end of the talk, but just wanted to jot it down right now. For the co-localization of pre- and post-synaptic markers is not being in the same 3D plane as the slice ever problem, aka co-localization is co-localized and perpendicular, as Zita mentioned. So basically, because all of these, because we're looking at these images from a microscope, and microscope uses light, and light isn't, like when you focus on a plane of light, it actually, there's a little bit of bleed through from above and below. You're saying, what if they overlap like this, but they're not really near each other, right? And we looked at that a lot, actually. That was really important to our work. Basically, the system that we're using is pretty good at that, and then we've tried to be as careful as we can about other ways that it can happen. Anything else?
I'll thank you regarding any specific method in the three-dimensional Harvard.
So you're asking about if you fill a cell and you can look at all of its connections and its dendrites, whether there's additional morphological changes or we just didn't look at.
More like a plasticity, like closer to.
Oh, yes. Also on the skipped slide, there is a discussion of that. Yes. Yes. So in short, that would be a really good thing to look at in the future. I hope for someone else, but maybe I'll have time to do it while I'm doing my postdoc, brief postdoc.
Beyond the scope of this talk question.
Yeah, beyond the scope of this talk. Haha, pun. Oh.
Okay, let's thank Derek.
So it's also customary to do a thank you. And while we're past the time, I do have the room for a lot longer. So I'm going to do it. And I think I'm going to have to restore it. Okay. 1 second on this. Oh, what's happening? Okay. 1 second. Alrighty. So I just wanted to say thank you in the traditional way for grad students at the end of this work. So first of all, we've joked about the dark side here. Wait, why is this not? 1 second. Okay. We've joked about the dark side here, and so I will continue to joke by talking about my mentors. I, a poor wannabe Kylo Ren, so lost, looking for his mentors. And Steve and Sacha have really been wonderful throughout this time. It's a little bit rare, they're not unheard of, to have a joint mentorship, but I think that it has been a really wonderful balance over the time that I've been here. I feel very well cared for and very well developed by the time that I've spent here with and under them. But of course, these are not my only two mentors. I also was mentored a lot by Vera, who was my direct postdoc when I came to the Nelson Lab, and by Yasmin, who was my graduate student mentor. For example, she's the one who got all of the calcium imaging system ready and working so that future grad students could work with it more. And actually, following up on that, also many of my conclusions and the way that my work is produced is based on her thinking. So big thanks to both of them. I also should mention my science antecedents, because I did not get to grad school without doing more work. I was in the Glenn Lab at Colby College, and that was a really good environment. And very nurturing and a lot of focus on how to do a good presentation, so hopefully we did a good presentation today, Melissa. And also at McLean Hospital, I had a great cadre of friends that I learned to do good cell culture there. You can see me suffering there in the middle. And I also now will turn to Brandeis, right? So the Nelson Lab has gone through many versions over the years. And I did a lot of my work in hanging out there because it's where my culture stuff was, even though I was in 2 labs. So I just want to thank everyone from the Nelson Lab from time past and present. Many great people and great scientists. And of course, the Van Hooser lab as well. Even though I didn't spend quite as much time there, they were still a big part of my life over the time. I want to thank all of the other people at Brandeis that I had really deep connections for. There are like 1 million people, so here's some who are like super important, and including my cohort of graduate students who have all generally moved on. One special thanks, though, to Chen Hao Liu, who started in the Nelson Lab at the same time that I did, and we sat next to each other. And I don't know, what a bright spirit and what a joy. And we've had a deep friendship for the entire time that we're here at Brandeis, and he's rocks. I also want to thank my D&D group. We talked about the nerd connection, and indeed, it is true. And so we started this a couple of years into my time at Brandeis when some people suggested to me that they would like it if I ran a game. And I did, and it has become a really wonderful social group and needed solved during the pandemic. I think we would all agree. It was very bad, but a lot better because of friends. So, also, not from the D&D group, but my friend Zeke is here, who came and played Magic with us in the Nelson Lab a whole bunch. So it was mentioned that I do historical European martial arts, and I actually started that here at grad school. Someone suggested the club that I'm now at because he was himself a member. And so it's been a really, as Sacha alluded, it's been a strangely parallel development between these two elements of my life, the grad school and also becoming master of the blade. So I want to thank Boston Armatari then. This is from our tournament over the summer that we ran. Many of the luminaries are there. But in specific, I also want to thank the triumvirs of Boston Armatari, Sean, Steve, and Mike is here supporting. I think maybe some others are remote, but I didn't check the full list of everyone who's there. And also my wrestling instructor, Ian, who looked like an egg in this picture. Also, I've hung out really a lot with, but unfortunately he has only this one blurry photo with Ethan, my good friend and sparring pal at BA. I want to thank the many pets, especially Nibs, who was our apartment cat. She's the best. And I also want to mention that I've, for a lot of the time that I was in grad school, but not all of it, I lived in Cherry Street, which is a long-lasting apartment in Somerville, with these fine lads, and I get to add an extra Nibs because it was on the poster. The last member of Cherry Street is not in this picture. He passed away while I was in grad school, but I miss him dearly. And I also want to mention specifically about a trio of pals, the Curtis family, who have been great friends since high school, or I guess even middle school. Many great moments and times and a great connection. I want to thank also my college and high school friends who are pictured here. I imagine there are many more, but these are some. And of them, I also want to make a special notice for Annalise and Hank and Marina. Hank was my first battle pal, where we would fight in the basement in the winter. And they have a beautiful baby now from a couple of years ago. And I want to thank Annalise for keeping me sane during the pandemic. And then for the past couple months, as I was putting together the thesis, I just wanted to mention a couple of people who have been incredibly generous with their time and energy. Chelsea and Mike and Allison and Vidya and Elizabeth, of course, from the Curtis clan, really went above and beyond the call of duty. And that happened also while I was at my beautiful summer house, which I think provided a lot of support as well. So it's in the slide. I'd also like to thank my 2 romantic partners of my life. I don't know, it's a strange thing thinking about whether you should, but I think you absolutely should. So thanks, Anna and Shauna. And I also want to thank my family, many of whom are here, for supporting me and bringing me up the right way. Bringing me up, clever, gotta do that. Here are some photos. And then finally, my grandmother passed away while I was at grad school. And that happened maybe 3 years in. But I think she made me who I am, so I wanted to show her a special painting. Okay, so that's done. And now it's time for me to run to actually pending, because we're a little late. So thank you very much for your attention. Thanks for letting me, thanks.